Okay. Okay. Taxonomy questions. First off, I want us to remember that you've got kingdom, phylum, class, order, family, genus, and species, right? And the domain is at the very tip top of that. So domain, kingdom, phylum, class, order, family, genus, and species. And remember that if you have two species, like let's say that the name of species 1, so let's just say name number 1, and then there's name number 2. Let's say that one species is, I don't know, Bulgari, simply, I don't know. And the next one is Bulgari, simply. Both of these have the same genus name. Remember, the genus name is always first, and then after that you have your species name. And if two members are of the same genus, then they're closely related, right? So they both have the same genus name, so they're closely related. I usually don't go over that. I think we got that for the most part. It's pretty simple. Since we're talking about taxonomy, let's look at maybe some of our more common kingdoms of life. Bless you. I think we would be looking at let's see, the common kingdoms would be like protist. So we'll talk about Protista real quick. We'll also talk about the plants, the fungi, as well as the animals. As far as some of the special features in each, for sure protists are usually living in aquatic or water environments. They're usually almost always single-celled. That's not always, but that's fairly often the case. For sure, all 4 of these are eukaryotic, so I don't think we should have a problem remembering that all 4 of these groups are eukaryotes. This, of course, is from the first chapter. There's a great little picture of it. The plants, of course, are multicellular creatures. Remember, they're doing photosynthesis. And does that mean that they're heterotroph or autotroph? Okay, so they're for sure an autotroph. Want to make sure you've got that. Let's see. What else? Having cell walls that are made of what? Cellulose. A fungus, though, is going to also be multicelled. But it's not photosynthetic. It actually has to eat things. It's actually a decomposer in many regards. So it's going to be a heterotroph. And it has a cell wall, but what is its cell wall made of? Yeah, chitin. And then animals are multicelled as well. Autotrophic, correct? No, heterotrophs. And no cell walls. Like you, no cell walls. Of course, you're an animal, so that makes sense. I've talked about this in an earlier video, but I think maybe we might want to just remember photosynthesis, like as an overall topic, and maybe just focus on the reaction that is photosynthesis for a couple of seconds. In photosynthesis, we know that we are for sure taking carbon dioxide in. We're also taking in water, and in truth, sunlight is also a reactant. And from that, we're able to make C6H12O6, and also some oxygen. Right? So there's kind of our overall reaction. Remember that the CO2, of course, is supplying the raw carbon. So this is the carbon that is used, or I could say fixed, in the Calvin cycle. And as it is fixed, it of course makes glucose. Of course, this is the result of carbon fixation, or the carbon dioxide fixation, or specifically reduction. The sunlight, of course, is supplying the energy, the raw energy, specifically for the light reactions, which include both photosystems, 2 before 1. Water, kind of the purpose of water usually gets thrown out the window. People kind of just disregard it, I think, a lot of times. Its purpose, though, is to supply new electrons. To the photosystems, right? And as an indirect result, that actually causes the production in an indirect way of ATP because of the buildup of the H plus gradient, and those it also produces the O2, which of course is the other product that we're seeing right there. So in a way, the O2 is a byproduct of the splitting of water, which is one of the first things that happens in the light reactions. Again, if there's some void here, I promise you that we've talked about it already. You just have to view the other videos. It's gonna be great stuff. Cool. We worked on a lab once about, and in an earlier video we had discussed osmosis, but we had worked on a lab once that had talked about diffusion and osmosis. Basically, we had discussed cell transport. And I want to go over just one part of that, that lab, or that experiment. It was actually part A of the experiment. And I don't know if you remember, but I had a beaker. And I filled it with just some normal water, H2O. And inside of that beaker, I put a piece of dialysis tubing. There's my dialysis tubing bag. Kind of just tied it to the edge there. And in the dialysis tubing, I had glucose, and I also had starch. And then into the beaker, I poured in some amount of Ki. Ki stands for potassium iodide. And kind of off to the side, I had made mention that when you have the iodine being added to glucose, it turns a real unique blue color. So again, whenever the glucose and the iodine come together, you have a blue color. And what we saw after a period of time was we saw that inside of the dialysis tubing, it actually did turn blue. So after about 30 minutes, one of the data points that we recognized was that the inside of the little container inside of the dialysis tubing, it was blue. And so was the water. That actually was showing us 2 events that were occurring at about the same time. One event was that the potassium iodide was able to enter the, what's it called? Enters into the tube, and glucose could exit the tube. The glucose test was positive in the water. So basically, your pores, or your holes in the dialysis tubing, were small, or were big enough for glucose and iodine, which are pretty small objects, but they were not large enough for starch, which is a huge compound. Cool? So I think you just want to understand, like, this idea of permeability is really dependent upon size. And that's what this lab was teaching, about how permeability, the ability of something to cross the membrane, is completely dependent upon the size that you have. Cool. Good stuff. Okay. Done. moving onward, I think we could spend a little bit of time just kind of reviewing some genetics problems. Yeah, I think these are pretty straightforward. I'll get you to write down some of these and then we'll discuss them.
What are we doing?
Record button again. Okay, I think we want to focus on functional groups for the millionth time. Why not? So let's go there. Functional groups. I think again you want to be looking at these functional groups multiple times. I'm going to give you functional groups. Hopefully you can keep up. C double bond O, C double bond O, H. C double bond with an OH, C with an N and two H's like that, C with an OH just like that, C with a PO4, and that's pretty good right through there, I think. I might think of one more that I want to throw in. Let's see, C double bond O in the middle of two C's, that's going to be a ketone. This one's going to be an aldehyde. This is going to be a carboxylic group, carboxylic acid, which goes great with the next group, amino group. Get it? Amino acid? Yeah. No, literally, those two are the functional groups in amino acids. COH, that's going to be hydroxyl, also known as an alcohol.
Oh, hey.
And there's a phosphate group, PO4. Usually that's representing a high or low energy. High energy, think of ATP. What about sulfate? Usually sulfate's not. Sulfahedral groups sometimes are. It would be a C with a CH bond, or with an SH bond, sorry, and that's going to be a sulfahedral group. Do we have that? Absolutely. On the exam? Nope. But we'll throw it in there. Why? Okay. Let's talk about.
So what are the questions like for functional groups?
Nope.
Are they just? They're like. You don't really have to know like the.
Like you have to kind of put the names together in the structure. Like for instance, they'll say that one of these guys, they won't give you the name at all. It would just be what you see in black, and they'll see formaldehyde is used to embalm bodies. Which of these is formaldehyde? Well, you'd have to know that this is an aldehyde group. Cool.
It's like the multiple
Will it
choice
have
gonna be
like
that picture?
Yeah, basically.
Alright.
But it won't have like an entire.
But it won't say aldehyde. They'll say they'll say butanone is a chemical that could be used to synthesize fragrances. Which functional group would you see in butanone? Well, one would be a ketone, so you'd expect to see this group right there.
Is it gonna have an entire molecule, and we're just gonna like find it on the end, or is it just gonna have those little like just a functional group?
You'll see the function. It'll just be the functional group,
Okay.
and maybe just a couple of other carbons around it, but nothing major.
So yes. Oh.
Stop, Bradley. You always do that to people that walk in here, and it really irritates me, dude.